Okay. So let's go with the exercise question. Here, the first exercise questions are related with the uh, genome browser data visualization part. So it's a task for you. I will just upload this PPT in the drive, or maybe I will just share the question in the chat box. You can copy paste somewhere for now. This is a same thing what we have already discussed yesterday. So please try. Whenever you are facing any doubt, any problem, I will guide you. But at least just once try this. Okay?
Okay, ma'am.
Yeah. Uh, Now, yesterday we were discussing about the table browser, correct? So first of all, I will give you some overview about the table browser, like what options and all we are having there, and then we'll proceed further for the exercise question. So here, this table browser is used for the data retrieval where you have to select the clade, mammal, vertebrates, whatever you want for which organism you want to retrieve. Genome, you have to select the organism here for which you wanted to retrieve the data. Then you have to select the assembly. Once it is done, next here you can see the group. Okay. So these are the same groups, actually, what we have already checked in the genome browser, right? You can see that mapping and sequencing, genes and gene prediction, phenotype and iteration, and many more. Correct? Then here you're having the track, means let us consider you have selected the genes and gene prediction. The track, you can all the tracks are coming under the genes and gene prediction group. You can select the track as per your choice according to which database you wanted to retrieve the information. Then table here, once the group and track is selected, you don't need to be select the table as table, like, because the table is automatically updated here. No need to change. Now, region, you want it for the genome, like a whole genome, or you want it for a specific position, like specific chromosome or specific gene. But the thing is, you cannot give here the name, you have to give the position only, location. Okay? And that location, it will be in a chromosome number, start hyphen end coordinate. Once this is done, next we are having the filter. Like these are the optional things for the subset, combine, compare with another track. Means if you have selected the NCBI, so if you wanted to filter something or you have to merge with any another track, correlate with any another track, these are some optional things. We are not going here right now. And next you're having the output format. So once you have selected all the options, in which format you want your output, like you want all the fields which are all the columns which are given by the UCSC by default, or you want a selected columns which is required for you, or you want a output in a sequence format, you can select. Then GTF, that's a gene transfer format which is limited up to the UCSC. Means this format you will get in a UCSC, for this IDs are limited actually for the UCSC. This is one kind of annotation file. I will explain you in the exercise question detailing, because right now even if I will tell you, you will not understand it, and you will not able to understand it. Okay. Then if you want a alignment, fast alignment, you want a bed format. Now this bed format is also browser extensible data format. This is also what is the bed format and all that also I will explain you during the exercise question. So this is what you can select the output format. And once the output format is selected, you have to give here the file name if you wanted to download the results in your system. But if you don't want to download the result, you just wanted to see the result, you can keep it blank. You can see add CSV extension if opening in Excel, leave blank to keep the output in a browser. You don't want to download it, you just wanted to check it, see it. So no need to give any name, keep it blank and then click on the get output. So here all the information is also given downside. If you want to check any another option, what it means, you can check it out here. So this is just a general idea about the table browser, but let's go with the exercise question so that you will get it properly. That retrieve all the refsec genes. Now, first and foremost thing you have to see that what word we are having at the starting, like a retrieve or visualize. Visualize if it is, you have to go with the table browser, sorry, genome browser. Retrieve if it is, you have to go with the table browser. Now, retrieve all the refsec genes along with a transcript, strand, exon region for first chromosome of human. What we have to do for the first chromosome of human, we have to retrieve all the genes from refsec. But along with that, you have to get the transcript information also, strand information also, and the exon information also. Question is clear?
Yes, ma'am.
Hello?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So here we are going with the table browser now. Clade is mammal. You have to perform here only. I will give you a time, first see and then perform. Okay. You have to go with the clade, genome human. Assembly is the current assembly at G38. Now group I am selecting the genes and gene prediction because the question is to retrieval of the gene. So genes and gene prediction and track, you have to select here the NCBI RefSec because as per the RefSec it is asked. Now no need to change the table region. So in the region, it is asked specifically for the chromosome 1. So you have to select the position and give the number CHR1. Okay. Once you have given the position, then the output format, I'm going to select the selected fields from primary and related table. Why? Because in the question, it is specifically asked to select this column only, not any another column. Transcript, strand, and exon. So that's what here, if you will go with the uh, like primary selected fields, you can select the columns which is required. And here I'm giving the name. You can give the file name as per your choice. .txt extension. UCSC result.txt extension. You can keep it CSV, TSV, whatever you want. And then click on the get output. So once you have clicked on the get output, as we have select the option in the output format selected fields, so it is asked to check the column which is required in our result. So what column we require? The name, right? Name of the gene. We require the chromosome also, to which chromosome it is located. We require the strand information as per the question. Then next it is giving the transcript information also, right? So transcript start and transcript end coordinate. Here you can see the CDS start and also there. I asked one question. I asked to solve one question for the UTR. Is it done? Like you tried that question or
No, ma'am, haven't tried it yet.
okay, no problem. So next you're having the exon count, and here this is what exon start and end coordinates you are getting. Score, actually, this is some statistical score. You don't need to select it. And the name too, I'm selecting here because in the first column, if you will see, it's a transcript ID and this is a gene ID, alternative name, like a name you are getting here. So I'm selecting the last column. And then you have to click on the get output. Once you will click on the get output, the result is going to download. Can you just try and tell me when it's done? What about others? Have you tried with the BRCA one, all the questions and the UTR question which I have asked to solve?
Yeah, I tried the UTR question.
Okay, so like any problem
No, I
or
could
is
find the, I just subtracted the CDS and
hmm,
found the 5 prime and the 3
hmm,
prime UTR.
hmm. Okay. So will you be like the FASTA sequence is retrieved properly or like
Yeah, because I had selected the show region. So
Change region.
yeah, selected
Oh.
region. So I just entered the numbers after calculating from the feature section, which one is the UTR region.
Mhm. Okay, all right.
Ma'am, in the table section, what we need to select no gene, right? Ma'am,
So yeah, it's done.
ma'am, can you show the table browser part again? Like,
Hmm.
what did you select it there?
Yes. Oh, sorry. Yeah.
Ma'am, the other one.
Table browser part. Okay,
Yeah,
okay.
ma'am.
Yeah.
Yes, ma'am, it's done.
Okay. So after this, what we have to do is we have to open that result, UCSC result in a Excel, because it's a column wise data. We can better see that in the Excel. So open that, browse, uh, select the all files, UCSC result, open, delimited it. Make sure you are doing it correct. Selected correct options like delimited it. Next, tab separated data only. Next, and finish. So next you just have to go with the file, save as, and save this in a Excel workbook format. First format, like you can see here, the Excel workbook you have to select and then save.
Ma'am, can you tell again how you converted it to Excel?
Yeah, you have to just go with the file, open, browse, Select the file here, open it, delimited it, next, tab separated, next, and finish. Then file,
Okay.
save as. You have to again grow with the browse, select the folder where you wanted to save it, and format should be Excel workbook. Okay. Just what you can do is create one folder by the name of Talk2Omics or LSSDC, whatever the course name, without the space. Make sure you're not giving a space to the folder name, and save all the file inside that, because right now we are going to create many files, many tasks. So it will be better for you if it is in a one folder. Okay. Yeah. Tell me when it's done. And every time, whenever we are making a changes, make a habit to save this. Done? Hello?
Yes, ma'am.
Yeah. What about
Yes,
others?
ma'am.
Yeah, ma'am, done.
Okay. So once this is done, next question, like this is how we retrieve, if you will see, like the first column here we are having is the name, chromosome location, strand information, to which strand it is located. This is a transcript, start position, end position, name of the exons, like how many, so exon count, like how many exons it is having, the three exons. So here you are having a three exon start, and here you are having a, all the three exon end positions, and this is a name. Now, one thing if you will observe in this, like some of the name of the gene, some of names in the gene column, they are repeating. Like if you'll see this CDK11B, this is repeating. Then here also, if you will see this LOC is repeating, this NPHP4 is repeating, AJAP1 is repeating. So any guess why it is like that? Any guess? Huh? Anyone?
Can you please repeat the question?
Hmm. Yes. Here, if you will see in the last column, the some names of the genes are repeating, right? That this LOC, if you will see, the same name is repeating here. Then, if you will go down here, somewhere, like this name of the gene is repeating. Here also, this name is repeating. So why it is like that? Like,
They might be alleles of a gene, like
but the ID will be changed, no? That time means. if you will see the first column, so first column is what? These IDs are what? Can you tell me? These IDs are about the transcript IDs, if you will see, correct? NM, NR, NCBI mRNA, NCBI non-coding RNA. So as this data is as per the transcript, one gene can form many transcripts. So if you will see, this LOC gene is forming this many transcripts, correct?
Yes, ma'am.
Got it? Any doubt here?
So, ma'am, it's like multiple ORFs.
Multiple transcripts. Yeah, ORF also, but not every transcript is a coding, no? Means
And ma'am, why there are like multiple coordinates of exon start and exon ends? Like,
because here one exon, this transcript is made up of exons, right? So let us consider this NM transcript which we are having. It is having a three exons. So this is giving a three exon position start. Like this is the first exon position start, second, third, and this is the end position.
yeah, ma'am, got it.
Hmm. All right. So let's go with the next question, that report the total number of genes on chromosome 1 of human. We have to report the total number of genes. So how can we report the total number of genes on chromosome 1?
Ma'am, we can remove the duplicates and count the number.
Exactly, because if you will see here in the first column, like we can see the total number of rows directly by control shift down. You can go directly at the end number. Here we are having the total number of transcripts, like 17,116 transcripts. But we want a total number of genes, and the total number of genes are present in the I column, last column. But here, the some names of the genes are duplicated, correct? Like this LOC and whatever I have discussed downside, names are repeating. We want a unique value. So what we can do is we can just copy the I column, like a name column. We can put it into the next sheet here. And in the data, if you will go with the data, we are having an option of remove duplicates. So if you will just click on it, select my data has header, because like our data is having a header. So you can select that, and name to column is selected, just say okay. So it is going to remove the duplicates. You can see 13,020 duplicate values found and removed. 4,096 unique values remain. Means total number of genes we are having is 4,096. One is for header, so that's why 96. This is what we are getting, the total number of genes. Is it clear?
Ma'am, can you show that again, ma'am?
Yeah, you have to put the you have to just copy the column, then paste it here in the A column, go with the data, go with the data, remove duplicates you have to select and say okay. Okay.
Okay, ma'am, got it.
Tell me when it's done.
Done, ma'am.
Uh, let's go with the next question. Retrieve, no, report the first gene of human and visualize it on genome browser. What we have to do is we have to report the first gene of human. Now, from this, which gene is the first gene and how can we retrieve, how can we report that? How can we report the first gene?
We could apply a filter which would show us the lowest value, like ascending order.
Can you just repeat once?
And we could apply the filter to the start one and select the lowest one.
Start, transcript start, you are saying, right?
Yeah, ma'am.
Correct. Like right now, maybe let us consider if you have a multiple chromosomes, maybe chromosome 1, 2, 3, 4. You have to sort the data based on the chromosome position. Along with that, you have to sort the data in ascending order according to the transcript start. Because shortest the transcript start, that gene will come first, right? So, as we have just for the chromosome 1 data, so we have to select the transcript start column.
Ma'am,
Go
why
with the
exon start?
exon start. No, no. Actually, exon start, if you will see, we have a multiple values here. So it is
Mhm.
not going to sort it properly, right?
Okay.
Because obviously, like multiple values, if they're so according to that, it is going to sort. So it's better that we have already the transcript start, right? Because the data is as per the transcript, correct?
Okay.
Hmm. So we'll go with the data, then sort. In the sort, we have to select the expand selection because along with the transcript start, rest of the columns also we want to sort it. So expand the selection, click on sort, select the value on the basis of which column you wanted to sort it, transcript start. Okay. Once this is done, you have to check this option also, my data has header, because as our data is having a header, it is not going to be sort then. So this option should be checked. Okay. Then order how we want, smallest to largest, right? Because in ascending order we want, and say okay. So it's already sorted actually, but maybe in future if you are not getting the sorted data, you should know how to sort it. So this is what the transcript start, smallest transcript start, and this is the first gene of human on chromosome 1, DDX11L1. And this gene we have to visualize it on genome browser. So we are going with the, oh sorry, I'm just going to be open this genome browser in a new tab. Yeah. So first what you have to do is you have to just go down and say hide all. Now as per the NCBI, we wanted to visualize it, make the NCBI full, and you can just copy the name from here. Sorry. You can just copy the name of the gene from here. TDX11L1. You have to just type, or you can just from here it is showing Homo sapien, or just a second. Better to type so that you can, it is giving directly the option. make the NCBR FSEC full, and here it is showing the third hit with the Homo sapien. Select it and then refresh it. Hmm. So, if you will see, like, this is no, this is the L9 I have selected. Sorry. Yeah, just enter. Uh, just a second, why it is not. Okay, so sometimes like here they have given the like here they have given the databases. So from here also you can select like NCBI RepSec genes, and here you are having the IDs. So this is on a chromosome 1, you can select the first ID. Hmm. And here, this is what a DDX11L1 we can visualize. NCBI RepSec mode is automatically pack actually. I don't know how it is updated. I'm making it full and refresh it again. So here you can see the DDX11L1 we can visualize. On which strand it is located? If you will see, it is located on forward strand. So obviously we are getting in a forward direction. Then here the dark blue patch, if you are seeing here, it's a exon. If you will keep your cursor here, this is showing exon 1 of 3, this is 2 of 3, and this is 3 of 3. Okay, like this is what a 3 exons we are having here, correct? The position is on chromosome 1. And how can you validate that really this DDX11L1 is the first gene or not? If you will just zoom out it, like after the zooming out, before the DDX11L1, we are not getting any gene. But after the wash 7P, you can see some genes are coming, right? But as before the DDX11L1, like no genes are coming, even if you are zooming it out, it means this GDX11L1 is really the first gene on chromosome. See, after the WASH7P, some genes are coming, but before the DDX11L1, like no gene is coming, which means this is the first gene of chromosome 1. Like whatever the data we are just checking, we are just whatever the information we are having, if you wanted to validate that information at that time also, you can use this genome browser. Okay, yeah, if you will see the numbers like DDX11L1, then next you are having is a WASH7P. The third ID is also same. If you will see, it's MIR6 something. See, but before the DDX11L1 there is a no gene. Got it? Uh, are you getting my point?
Yes, ma'am.
And can you show how you reached to the DDX11L? I'm not able to get it.
Yeah, actually,
What is
what I
it?
did is I make the NCBI RefSec full. First of all, I hide all the thing. Then I have make the NCBI RefSec full. I refresh it. Okay. Then I put the name, click on search, and it is showing the database's name. Maybe you can share your screen once. I can guide one more time. Is it fine?
Yeah, ma'am, I will share it.
Yeah. Refresh it first before remove the DDX11L1 for now. Okay, go down. Make the NCBR FSEC full. Make the NCBR FSEC full. Refresh it. Now type the DDX11L1 on in the search box. Now see, whenever you are doing the hide all now, automatically it is going to hide the NCBR FSEC also. So no problem. Just make the NCBR FSEC full, refresh it and type the name directly. Type the name or just paste the name here. Search, click on search. Okay. Now, if you will see, no, not this, not this. It's a Hugo gene nomenclature. You have to go with the NCBR. This is a RefSec genes. The upside is a NCBR RefSec. Yes. Open that. Click on the ID. Yeah. See, now make the NCBR RefSec full again and refresh it. Because this is pack. Yeah. Okay. Is it clear to everyone?
Yeah, ma'am, got it.
What about others? Yeah, Juhi ma'am, is it clear?
Yes, ma'am.
Okay. What, Sania ma'am, yes, sir, Pushpit sir.
Yes, ma'am.
Okay.
Yes, ma'am.
Namrata ma'am. Yeah. Okay, fine. All right. So, yeah, please stop the screen sharing, sir. Let's go with the next question. Report, retrieve the FASTA sequence of first 10 transcript. What we have to do is we have to retrieve the FASTA sequence of first 10 transcript. Now, to retrieve the FASTA sequence, if you will see, we have a start and end coordinate for first 10 transcript, right? And now, if you have a chromosome location, start and end coordinate, you can just go with the table browser, and here you can give the position like CHR1, then colon, whatever the start coordinate and end coordinate, right? Likewise, this is how you can give. But as it is asked you to retrieve the FASTA sequence for 10 transcript, at that time it is quite tedious to give the positions one by one, right? Because right now it's a 10, so maybe you will be able to do it. That's fine. But in case, just consider that if you have uh, more than 10 transcripts, like 20, 50, at that time it is quite difficult one to do it one by one. So what I want to do is I wanted to retrieve all the 10 transcript sequence in a one go. Okay. So for that, are you getting my point? First of all, what I want to say?
Yes, ma'am.
Yeah,
Yeah.
ma'am, got it.
So I'm going to select all the 10 coordinates. First of all, let's copy, and by adding a one sheet, I'm going to paste it here. All right. Then they are located on chromosome 1. So I'm just going to copy the position of chromosome 1 also. And then here I'm going to paste it. I'm not giving any header or something right now. This is the chromosome location. This is the start coordinate. This is the end coordinate. So this format, we called it as a bed format. I've told you that I will tell you this bed format later. So bed format, browser extensible data format. This is nothing but it's just a chromosome location, start coordinate and end coordinate. Okay? Is it clear? Now I'm going to copy this bed format. You have to go here with the UCSC, and you have to go here with the My Data custom track. In the My Data and custom track, Here you have to paste your bed format and you have to submit it. Once you have submitted, here if you will see, it is showing you genome browser, like 10 items selected. We have selected the 10 positions from the chromosome 1. So it is correct, means like our data is properly submitted. But view in, we don't wanted to visualize it, we wanted to retrieve it. So I'm just selecting here the table browser. Okay. Once you have selected the table browser, you have to click on the go to first annotation. Can you try it till here and tell me when it's done?
Done, ma'am.
Okay.
Yes, ma'am, done.
Okay, so now once this is done here in the group, the custom track is selected, no need to change. Track is user track, no need to change that here. Table is also no need. In the position, you have to just give the CHR1, chromosome 1. Okay, because it's from the chromosome 1. And then which output format I have to select here now?
Bed format.
No. What is the question? Read out the question properly.
Oh, faster.
Right. You have to select the sequence, correct? Then I don't want to download it for now. I'm going to keep it blank and then get output. Click on the get output. Get sequence. And this is what you're getting the faster sequence. So if you will see the position now, just a second. Can you tell me whether the position is matching or not? You can see here the position is given CHR1, start position, end position. Here also. So whether it is
Ma'am, it's
matching or not.
873 and it's there is 874.
Hmm. And what about end?
Yeah, yes, ma'am, it's same. And number
Okay.
is same.
Yes, right.
Course,
What about others? Like, are you getting her point?
Yes.
Yes, ma'am.
What's the problem here? If you will see the start position in the UCSC, there is one extra number because in our input it's 11873. Here it's 11874. Actually, there is a drawback of UCSC whenever we are giving the input of bed format. UCSC automatically added one number at the start position. So it is going to be subtract a one base. Like it means it is subtracting or it is uh, like one base less sequence we are having, which is not a correct sequence what we want, correct? So this is not a correct sequence. What we are going to do it then? We are going to already subtract a one position from the start coordinate and then we are giving as an input to the UCSC, so that UCSC will add a one position at start, and we are getting our actual results. Why I didn't show you this previously? Because I want to show you the drawback. Because if I will directly subtracted one, you are not able to understand that why we are doing this, right? So that's what now we are going to make a correct build format. I'm going to copy this as it is, paste it here, and I'm going to subtract it one from the start coordinate. So this is a start position minus one. If you will select this, then here you are getting the plus symbol like this. If you will keep your plus symbol here at the end, and if you will double, okay, like as we have a one gap in between, so that's why it is not applicable. So you can drag it. Okay, once you have drag it here, this is what we are getting, the one value less. Can you try till here? Is it clear like why we are doing this?
But ma'am, I didn't get the point. Like, why are they doing that? Like, why are they doing?
How I can also like when I also don't know the thing. This is a drawback or something like.
But ma'am, there might be some reason. Like, if they are making such complex website and all,
Mhm.
and they are not, then they are doing this kind of stuff. So
Are you get like I'm getting your point, but that's the thing now means like right now we are having such kind of results, but we need the actual positions. Maybe there is some reason behind it. I'm getting your point, but as we want the actual sequence, the correct sequence for our position, so this is how we can get it, right?
yeah, ma'am, got it.
Yeah, maybe somewhere it's need there's a need of like one base less. So that's why they are doing that. Your point is absolutely correct, but um I don't know the reason behind it as
Actually, I think the reason is many servers use masking data, so they mask out some regions. Like in Nextcloud, they use the first and last region to be masked out.
actually so you're not audible properly. Can you repeat it once?
Now, actually, I feel that some web servers use the mask data, so they completely hide out the first or last region, and so they change many coordinates. Like in Nextcloud server, they use for
Hmm.
monkeypox.
Yeah, right. Yeah, this can be the reason also. Correct. Okay. So yeah, this is done. If you will see here that we are having the formula means like we need this value here in the J column, but the formula is present, and due to the formula, if you will copy paste, the values are not going to be pasted properly. It is going to change. So we have to remove the formula from, right. So how to remove the formula? You have to select the values, like you have to select the data, copy the data. Once you copy, right click, paste to special, and in the paste to special, you have to select the values. Okay, and say okay. Here also you will get it. Paste to special, and here also you will get the values. Here also you can click. And now if you will see, there is a values only, there is a no formula. So here you can copy this, select the first value and paste it. So this is what your correct bed format, correct values you can say. Then we just have to copy this, go back to the QCSC, uh, my data again, custom track. Then you have to add custom track, click on the add custom track. Then you have to paste it here, submit it. Then go to first annotation. Rest of the things will be same. Here you have to make it the position chromosome 1 only, and then output format sequence, get output, get sequence. So now you can see this is what a correct position you have now. Done?
Yeah, done, ma'am.
What about others?
Doing, ma'am.
Okay, no problem. Take your
Uh,
time. Yeah,
where did you select the FASTA sequence? Because that's the
output format sequence you have to select in output format.
okay.
Yeah, yeah. Here. Can you see?
Yes, ma'am.
Hmm. So this is that's it for today. We'll discuss this rest of the question by tomorrow, and we'll start a new database that's a PubMed by tomorrow. Okay.
Okay, ma'am.
I hope everyone is joining from the LMS portal, correct?
Yes.
Okay. Yeah, because I'm not going to share the attendance link from now, as I have discussed earlier. I think some like one of you like it's facing a issue from joining. So is it resolved now?
Yes, ma'am, it is sorted now.
Okay, okay. I think someone else, Sonali ma'am, like
Yeah, it's just that I'm not getting the recorded videos. Now I'm able to join. That's not a problem. It's just I'm not
In
getting.
the recorded, like from where you are joining now, on the right side also only there is an option of recorded video. Recorded videos, as in like every day you will get the videos, video like YouTube link now. Means previously how you are getting the recording from the drive. From now you are getting the link of YouTube videos there. On the right side you can just explore it once and
Okay.
see.
Yeah.
Okay.
Yes.
Okay. All right then. That's it for today. Rest of the things we'll discuss by tomorrow.
Okay. Thank you, ma'am.
Yeah, you're welcome. Bye.